Hello, my name is Ravinder Abrol, and I'm from the Department of Chemistry and Biochemistry at California State University Northridge, where we study membrane proteins like GPCRs to understand their folding, structure, and signaling. I would like to thank the organizers for giving us this opportunity to talk about our work, which was carried out by Bridget Kavamala, a bright student in the lab, shown here. The motivation for this project came about when we were doing a collaboration project with Michel Bouvier at Montreal, where we were looking at a GPCR called vasopressin receptor, which shows mutations in patients, and these mutations cause these proteins to misfold, so they're unable to escape the quality control of the ER. However, they can be rescued by pharmacological chaperones that stabilize these receptors and enable them to be transported to the plasma membrane for their normal function. Our goal in this project is to understand how membrane proteins fold and how pathological mutations can cause them to misfold. These proteins pose a major biophysical challenge because they are stable only in a membrane type environment. They also exhibit a huge number of structural folds as shown here. Three of the most common topologies shown for membrane proteins are in terms of being alpha helical or as beta barrel. And our focus is on alpha helical membrane proteins, which will have about 7 or more domains. When these domains come together, they actually form this helical bundle that looks like this in 3 dimensions. In order to study how these proteins fold, about 30 years ago, a two-stage model was proposed by Popot and Engelmann, where in the first stage, the helices are formed and stabilized in the membrane, and in the second stage, these helices come together to form helical bundles. Over the years, this model has been enriched with many studies, here envisioning how helices might enter the membrane from the translocon, and here where the helices might enter the membrane independent of the translocon as well. In addition, helical hairpins, shown here on the right, are tertiary structures that have been found to be important for membrane protein folding, and they can insert into the membrane independent of the translocon as well. In the last couple of years, several studies have come about that have shed some light on how these helices might get inserted into the membrane. The study on the right shows a sequential insertion from the N-terminus helix to the C-terminus helix, whereas the one on the right shows a random N to C-terminus order of insertion into the membrane. In addition, over the last few years, the role of the membrane-water interface is emerging to be an important factor in how these proteins fold. So we have been looking at this process by reconstructing protein folding models in terms of these different compartments that the helical segments have to go through. So in order to understand the major features of their folding pathway, we first wanted to analyze available helical membrane protein structures and see what we can learn from them. So we collected about 246 curated helical membrane proteins from the MP-Topo database, and we did a hydropathy analysis using the White-Wimley octanol interface scale that gives us, with the area under the curve, it gives us the free energy of insertion of that helix either at the interface or in the membrane environment. These 246 proteins, they cover a wide range of sizes from 1 TM domain all the way to 24 TM domains. For each size, one protein is highlighted here. And here are the hydropathy plots for representative proteins. In the interest of time, I won't show you all 246 hydropathy plots. The area under their curves averaged over these proteins are shown here, which did not show any clear pattern. Then we looked at how the helix insertion energy into the interface behaves in terms of the helix insertion energy into the membrane. And here's an example showing for the protein family that has 12 TM domains. And when the data is plotted, it shows that the interface energy is about half of the membrane stabilization energy. And this curve looks remarkably the same independent of the size of the helical membrane protein. And we get a slope of about half, and to be more precise, we get a slope of about 0.46. So that tells us that the interface stabilization of the helices is about 46% of the total stabilization they will get when they are inserted fully into the membrane. In addition, we also get an additional 1 kcal stabilization of energy per helix from this data. So this proposed to us that the interface probably plays an important role, and we created this three-stage model of membrane protein folding where the interface acts as the intermediate stage. And to test this idea, we looked at two membrane proteins and we looked at their most hydrophobic domains along with one of the proteins that also exhibits a helical hairpin called the paddle domain. And we subjected each of these domains to 4 microseconds of MD simulations in explicit membrane environment. And in addition, each of these domains was oriented in 3 different ways in the membrane, either at the interface, which was parallel to the membrane, or in the middle of the membrane in terms of parallel or perpendicular to the membrane. And here's a summary of 48 microsecond MD simulations, which shows that when the transmembrane helices start at the interface, shown in blue, they stay there, which means that these very hydrophobic regions are quite stable at the interface. However, if they start at the middle of the membrane and parallel to the membrane, shown with green, they are stable there or they go to the interface. However, if they start in the middle of the membrane in a transmembrane orientation, then they are stable there. So this showed us that the interface could be a viable vestibule where the helices could spend time before they get enough energy or before they are able to go over the kinetic barrier to get into the transmembrane orientation and get further helix stabilization to be inserted into the tertiary fold. Thank you.